Ever thought about following a recipe, not for a cake or anything, but for creating something like life-saving insulin or a powerful new vaccine? Well, believe it or not, that's pretty much what scientists in biotechnology do. It's called recombinant DNA technology, and it's basically a high-tech recipe for making amazing things. So, let's get our lab coats on and break down exactly how it works, step by step. Okay, so here's our game plan for today. First, we'll get the hang of the basic recipe itself. Then we'll grab our main ingredient, the DNA. After that, we get to the really cool stuff, how to cut, paste, and make billions of copies of it. Then we'll pop it into a living host, scale it up in these huge things called bioreactors, and finally, put on the finishing touches. It's the whole journey, from a single gene to a finished product. So at its core, what are we really doing here? The whole idea is kind of like a genetic mashup. You're taking DNA from two totally different places and sticking them together to make something new and super useful. Imagine taking a human gene, let's say the one that tells your body how to make insulin, and putting it inside a simple bacterium. That bacterium then becomes this tiny, living factory, just churning out human insulin for us. That's the magic right there. All right, first things first, any good recipe starts with prepping your ingredients. And for us, our star ingredient is pure DNA. Now, the catch is, DNA doesn't just sit by itself. It's locked up tight inside a cell, all tangled up with other stuff like proteins and RNA. So our very first job is basically a smash and grab. We've got to break open those cells and pull out the pure DNA, leaving all the other junk behind. And how do we break those cells open? This part is really clever. We use these tiny biological tools called enzymes, and it's like having the specific key for every type of lock. If you've got a bacterium, you use an enzyme called lysozyme. For a tough plant cell with its cellulose wall, you bring in cellulase. And for a fungus with a chitin wall, you guessed it, you use chitinase. Once we're in, we unleash a couple more enzymes to dissolve all the unwanted RNA and protein, leaving just the good stuff. So, after all that chemical cleanup, this is what you get. You pour in some ice-cold ethanol and, because DNA can't stay dissolved in it, bam, it crashes out of the solution and clumps together. You can actually see it with your own eyes, these fine, gooey, whitish threads. That right there, that's our pure genetic material, ready for the next step. Okay, we've got our DNA. Now we get to what is honestly the coolest part of the whole process. This is where we become molecular surgeons. We need to go in with absolute precision, find the exact gene we want, snip it out, and then make millions or even billions of identical copies. Our main tool for this job is something called a restriction enzyme. And the best way to think about it is as a pair of molecular scissors. But these aren't just any scissors. They don't cut randomly. Each type is programmed to find a very specific sequence of DNA letters, and snip, it cuts right there and only there. It gives us incredible accuracy. Great, so we've snipped out our gene, but one single copy isn't going to do us much good, right? We need an absolute ton of them. And that's where our next gadget comes in, the polymerase chain reaction, or PCR. If restriction enzymes are our scissors, you can think of PCR as our high-speed molecular photocopier. Its only job is to make copies of our gene. So how does this molecular photocopier actually work? The whole thing is driven by a simple cycle of heating and cooling, which has 3 key stages. Now, don't worry if this diagram looks a little technical at first. The basic idea is actually beautifully simple, and it just repeats itself over and over and over. Okay, here we go. Step 1 is denaturation. We crank up the heat to about 95 degrees Celsius, which makes the 2 strands of the DNA double helix unzip and separate. Then, we cool it down for annealing. This allows these tiny little starter sequences called primers to find and stick to the beginning of our gene on each of the separated strands. Finally, in extension, we heat it back up a bit, and an enzyme called DNA polymerase starts building a brand new matching strand. And just like that, 1 copy has become 2. Now, turning 1 copy into 2 is neat, but here is where the real power kicks in. You just repeat that cycle. So 2 copies become 4, 4 become 8, 16, 32. You see where this is going. If you do this cycle just 30 times, which only takes a few hours, that one single gene gets copied into about 1 billion versions. Yeah, mind-blowing, right? That's exponential growth for you. All right, so now we are practically swimming in copies of our gene. So what's next? Well, these gene copies can't do anything by themselves. We need to put them to work. We need to get them inside a living factory, a host organism that can read the gene's instructions and start cranking out the protein we want. Usually, the go-to host is a simple bacterium like E. coli, mostly because it grows incredibly fast. We use another enzyme to ligate, that's just a fancy word for paste, our gene into a tiny circular piece of DNA called a plasmid. Think of the plasmid as our delivery vehicle. Then we treat the E. coli cells to make them competent, which basically just means we make their cell walls leaky for a second, so they'll suck up our plasmid right out of the water. But here's a little problem. How do you know which of the millions of bacteria actually grabbed our plasmid? Most of them won't. This is where scientists use a really clever trick called a selectable marker. When they build the plasmid, they put in our target gene and a second gene right next to it, usually one that makes the bacterium resistant to an antibiotic. Then they just grow all the bacteria in a dish that contains that antibiotic. And what happens? All the bacteria that didn't get our plasmid, well, they die. Only the ones that were successfully transformed will survive. It's brilliant. So, success. We've got our genetically engineered bacterium and is working. But, a few successful cells growing in a little petri dish isn't going to cut it. We need to make enough of this stuff for the whole world. It's time to scale up. Way, way up. It's time for mass production. And this is the million-dollar question in biotech, isn't it? How do you create the absolute perfect conditions to grow trillions of these cells so they can produce massive amounts of your product? Well, the answer is a big, fancy piece of equipment called a bioreactor. And here's a look at two common types. On the left, you've got your standard stirred tank bioreactor. It's basically a giant, highly controlled stainless steel vat. But check out the sparged version on the right. By bubbling sterile air up from the bottom, it massively increases the amount of oxygen the cells can get. And more oxygen means happier, more productive cells, which means a much bigger yield of our product. Let's take a quick look under the hood. You've got a motor turning an impeller to keep the culture broth, that's the liquid with all the cells and nutrients, mixed up. A foam breaker to keep things from getting too bubbly, that sparger is pumping in oxygen. And the whole thing is loaded with sensors to control the temperature, the pH, everything. It's basically a 5-star luxury hotel for our engineered microbes. Okay, so after a few days in their luxury hotel, we have a massive tank full of culture broth that contains our protein. But we're not quite done. Our protein is still mixed in with all sorts of gunk, leftover cell parts, waste products, old nutrients. The final, critical step is to separate and purify our product. This whole cleanup phase has an official name, downstream processing. It's a whole series of purification steps to get our protein isolated and make sure it's incredibly pure. And for anything that's going into a human, like a medicine, this stage is absolutely crucial. They also have to get the formula just right, add preservatives, and then it has to go through tons of rigorous clinical trials to prove it's safe and effective. And that's our recipe, from start to finish. So let's do a quick recap. We started with DNA isolation to get our pure ingredient. Then we did cutting and amplification to snip out our gene and make billions of copies. We put it into a host to create our little factory, used a bioreactor to scale it way up, and finally used downstream processing to get a pure, finished product. When you really think about it, what we've just walked through is so much more than a technical process. It's a fundamental recipe for editing the code of life itself, allowing us to make these incredibly valuable products. So that leaves us with the big question, right? Now that we have this recipe, what else could we cook up? What are the next cures, the next fuels, or the next foods we're going to create? The possibilities are really, truly endless.